Is there any questions? Does anybody have any questions? If not, let's go over these guys quickly. Last time we left with cube jelly, is that right? Cubozoa class. Cubozoa, they're the most venomous animal on planet Earth. It depends who you talk to, I guess. And as far as the structures in here, nothing you have to worry about. Here are some pictures that these guys, in general, jellyfishes, can inflict human, and they have long tentacles. They can bring food to the mouth. And here's a little kid who was slashed by jelly, either cube. I doubt it was cube jelly. It was other type of jellyfish, and then not pretty. So they can eat other animals, like this one. This jellyfish is eating a crayfish by their long tentacles, so on and so forth. And other animals can eat jellyfish. I don't think this jellyfish is planning to eat the turtle. So if you saw it on the video, so other animals can eat jelly. Jellyfish can eat other animals. One question one student asked, and he said, Amir, as we are going along, is that true to say that primitive animals are eaten by more advanced animals and not the other way around? I said, no, that's not true. In this picture, if you look at it, your textbook have some pictures. A crayfish was evolved after jellyfish. If we look at the evolutionary tree, sometimes your textbook has a fish, a little fish that jellyfish is eating. Your textbook, or hydra, you saw it on the video, they're eating. And hydra is more primitive animal than a fish. So that's not true to say primitive animals are food for more advanced animals. No, it can be the other way around. Anyhow. Class Anthozoa, the last class, the common name for these guys is the sea anemones, not the entire class. The common name for the entire class is not flower animals, but the sea anemones, when we go to Monterey, there are some beautiful, gorgeous, colorful animals. They are bright green and bright yellow I've seen, and bright orange. These are gorgeous looking animals. We do not have it in the lab, but we do have sea anemones in the lab for you guys to look at. We have them for dissection. If you want, I'll take them out today so you can dissect them today. We have one in those containers, in those blue and orange containers that have been dissected for you. There are labels in there, but the dissection sea anemones are not going to be in the exam. We do have a model of it. I should bring the model and go over the model of the sea anemones. Here is the sea anemones. And what I was talking about, the flower animals, these tentacles on top, right here, they cut the animal and show you the inside of it. Simple animal. But these tentacles are colorful, very beautiful color. Bright orange, bright yellow. And I've seen them in Stein art, when it was the old Stein art. In Monterey, they are less, but there are some nice ones maybe in Monterey if you look at the displays. And sometimes the body, the trunk of the animal is also colorful and nice and beautiful. The whole animal is bright orange, bright yellow, other colors. Yes, what is it?
That'd be radial symmetry.
This is a radial symmetry animal. Still, we are talking about radial. I haven't left radial symmetry animals yet. Oh, I'm going to leave them soon.
Okay.
I'm going to leave them, go to bilateral symmetry animals, but still radial symmetry animals. Any way you cut this animal, any way you cut it longitudinally, it is same. External features are same. They're all marine, okay? We do not have any freshwater ones. And pharynx or gullet is present. So on these animals, this area right here, the middle of the animal right here is called pharynx or gullet, okay? So, and they have polyp stage only, and the medusa, they do not have any medusa. They do not have any medusa stage. And tube anemones and thorny corals, they're all tube anemones. Yes, I did not, in your PowerPoints, guys, I did not mention these are different type of corals. You do not have to know the name of them, but just know they're corals. These are all different type of corals. Okay, so and we have the common name thorny corals. They look like human brain. Okay, so and they all have different names, but don't worry about them. I don't know them all. There's so many different type of them. We have even more variety of corals in my collection. Gastrovascular cavity have septum. So here are, if you would, they cut the animal longitudinally, and this is a complete septum. So the septum separation, you have it in your nose. This is a septum in your nose right here, separates the right side from left side. Your heart has a septum, separates from your right side from your left side of the heart. So you do have septum in your body as well. Not everywhere, but some. But these guys have 6 complete septum. That's what they are. And then inside you have these incomplete septum. Right here, if you can see it, that is this incomplete septum, the smaller one. So that's pretty much what a gastrovascular cavity is separated. Mesoglia has amoeboid cells. The mesoglia layer here, it would be right in this area, that has the amoeboid cells. Here is a cross section. As I said, there are 6 complete. There are 6 of them. 1, 2, 3, 4, 5, 6. This is the pharynx, gullet or pharynx, and these are incomplete septum. Okay. Again, that's pretty much all about the nematocysts are right in this area. Okay. So they do have them. Gastrovascular cavity here, gonads, acontia is a structure. You do have it on for locomotion, for trapping food, and so on and so forth. Pedal disc, the bottom portion of the animal is called pedal disc. Okay, and that's it. The tentacles on top. There's not much septal perforations with these openings right here called ostia, which the water percolates in them, and so on and so forth. Again, you can read about it, or we can talk about details of them, what these animals are all about. But again, we have to talk about other things. But quickly, different species, not metridium that you have in the lab, they're like this, the anemones, and they make these calcium carbonates. Okay, they make these calcium carbonates, and they put them on the side of the animal. So it would be like this, these calcium carbonate that I showed you. It would be here, and then when the animal dies, the soft portion of the animal dies, these calcium carbonates will remain millions of years. That's what you're seeing, the corals, or reef coral you've heard about. So they make reef. As I said, when a ship Titanic, a ship sinks, that's a reef. When they make these calcium carbonate deposits and they die, then these calcium carbonates become a reef on the bottom of the ocean, on the bottom of the sea. I hope I'm making, so you hear the term coral reef. That's what it is. You have the bottom of the ocean is straight, something comes on top of it, then it forms a reef. It forms a plateau. Corals is forming a plateau. A ship, a shipwreck that sinks on the bottom of the ocean, or a submarine. Now you have a reef. These are reef. They make a reef. Whatever, volcanoes, whatever comes up that makes the bottom of the ocean a plateau, they have a reef. And then of course on reefs, these are not just reef, they're plankton, algae grows, and other animals come and eat those algae, and the food chain, and so on and so forth. So they are not just there, they do house other animals, those corals, they're not just there. Okay, then I'm all done with phylum cnidarian. This belongs to phylum cnidarian. What class? Class Anthozoa. What one particular species that we have in the lab is Metridium. Metridium. Okay, so make sure you look at the specimens we have. The other phylum, we do have this one in the lab also. One species of it, the comb jelly, when we go to Monterey Bay Aquarium on 26, is that right? On 26 is a Thursday. When we go there, be here around 7 o'clock, not around 6:45, let's say. I'll be here, I'll drop off my kid at 7, and then I'll be here at 7:05. Don't call your classmates, oh, Amir, Farhana is in the car, can you stop the bus? No, let's not do that. Please, just be here. We are not going to stop the bus after we take off. Okay, or can you wait, Amir, 5 more minutes? Jose is coming. No, at 7 o'clock we have to take off. 7:05, 7:10. As soon as I get here, we have to take off. Okay, example of pleurobrachia. Yes, we do have in the lab. This organism is in the lab, pleurobrachia. And Ctenophora resembles cnidarians because these are the similarities. They resemble. These are the similarities between jellyfish and comb jelly. Okay, now I'm talking about comb jelly. They're radial symmetry animals. They both are radial symmetry animals. Comb jelly and jellyfish, they both are radial symmetry animals. Okay, they have oral axis. Aboral and oral axis. You know jellyfish. Okay, this is the oral side, mouth and anus, quis possibly. These animals have the cnidarians, mouth and anus, same place. Do that make sense? Mouth and anus, same place. Okay, cnidarians. But these guys, no, you will see that. But they have, since they are they're radial symmetry animals, they have oral and aboral. Okay. Well-developed mesoglea layer. You all know that. No coelomic cavity. No coelomic cavity does not mean they are acoelomate animals. That's why I disagree with your textbook, your lab manual, or anybody else that say acoelomate animals do not have coelom. They do have coelom, but the coelom is filled with nerve plexus. They have nerves, lack organs and systems, just like cnidarians. These are similarities between cnidarians and these guys, the Ctenophora. Okay. Now, why they have to put them in a different phylum? This is the answer. Why they have to be in a different phylum? Aside from DNA differences, no nematocysts. They don't have that. So they have to go to a different phylum. Distinct muscle cells. They have, you remember, the cnidarians had muscle fibers attached to epithelial cells. These guys, they have distinct muscle cells. They have cell membrane around them, around the muscle fibers. Okay, and they have comb plates. They usually generate electricity. When you go to Monterey, you will see those comb plates. They light up. Okay, when you see them on a display, you would know you have a comb jelly on hand. That's what they call them, comb jelly. No polymorphism or dimorphism at all. They're not colony. They do not have medusa polyp stage. That's not just all medusa stage. That's it. The egg becomes medusa. And never, of course, never colonial. I talked about that. And they have anal pore. The anal pore usually is on the aboral side of the animal. Okay? They have anal pore, unlike cnidarians, which they do not have anal pore. And those are pretty much all of everything that I need to say about cnidarians and sponges. Let's close this up and go over acoelomate, coelomate. Yes, right here. Okay, so we did today briefly. We talked about this. Eric talked about it a little bit, but let me go over it. The difference between acoelomate animals, pseudocoelomate animals, and eocoelomate animals. Quickly, let's go over it. Coelom, by definition, is a space. Quick definition, quick, dirty definition of coelom is a space between gut, right here, and epidermis. That's a quick, dirty definition of it. But the definition is longer than that. The space between gut and epidermis that is surrounded completely, that is surrounded by mesoderm. Okay? If you remember that, then the rest should be easy. That is surrounded by mesoderm. So here's a gut, here's the epidermis. Gut came from where? It came from endoderm. And epidermis came from there. Epidermis, where is epidermis? There is no epidermis? Yes, I guess. Epidermis, they didn't label it. Epidermis came from where? Ectoderm, right? We already learned that last exam material, or a couple exam material before. Now, in between, the space in between, look at it. It's completely surrounded by mesoderm material. So that's why they call this, that's why they, not us, is that right? That's why they call this acoelomate animal. A means without coelom, but really they do have coelom. The coelom is filled with mesoderm material. Look at pseudocoelomate. Pseudo means what? On your prefix suffix, pseudo means what? False. Okay, so you have epidermis, which came from, did they mention it? Ectoderm. It came from ectoderm, and the gut came from endoderm. Now, then you have an empty space. And on your slides, you will see that. On microscopic slides from true animal. They got an animal, they got a roundworm, and they cut it, and you look at it on a microscope, you will see that space. Okay, that space is surrounded by one end, is surrounded by mesoderm, and the other end is surrounded by what? Endoderm. So that's what they call it, pseudocoelomate animals, false coelom. This space right here, look at it. This space right here, all of it is surrounded by mesoderm. So this is an eo. Eo, it means what? True coelomate animals. Ida, is that right? Am I? Ida? Huh? Ida, am I right? I have an idea. English language is not good. I tell them it's very hard, very difficult. They don't believe me. Okay. So now you know the difference between eocoelomates, pseudocoelomates, and of course this exam material, you do not have these two in your exam. But exam number 4, eocoelomates and pseudocoelomates are going to start coming to a picture. But for final exam, you should know what animal is what, acoelomate, eocoelomate, and of course, cnidarians and sponges are they're not even here, right? They're not none of these. Sponges, cnidarians, ctenophora, none of them. Maybe one of the reasons because they're what? They're diploblastic animals. They're diploblastic animals. This only talks about triploblastic animals. I hope I'm making some sense. Everything coming to. Okay, the acoelomates animals, quickly, let's talk about them. Some cephalization, and of course their bilateral symmetry. First group of animals you're studying that they are bilateral symmetry. First group of animals. Platyhelminthes, or flatworms. Platyhelminthes, Ariana, is that right? The tolerance for my caffeine is going up. 1 cup a day is not enough. I gotta have more and more. Soon I'll be drinking Red Bull. Is that right? Red Bull got sued because somebody jumped from Golden Gate? That's what my daughter was telling me. It says you have wings on it. Is that true? Am I making it up or is it true?
I do believe they got sued because
There is a lawsuit.
people did not grow wings.
And that's it. That's what my daughter was telling me. I said, no, that's not true. I didn't believe her. But we were in Home Depot and in a cooler it says wings. You grow wings. And I said, Daddy, see, I told you. Okay, I don't know these things. She knows things that I don't know. God, help. But anyhow, so my tolerance for caffeine, I guess I gotta drink more. True organs. First group of animals. Something you remember I said for your final exam, I will ask you general characteristics of each phylum, Well, of course I said here is the acylomates, but we're talking about platyhelminthes. The only acylomate you're going to study is platyhelminthes. They have true organ now. Other animals we studied so far, no true organ. Protostome and spiral cleavage, of course, that's a given characteristics from beginning of simplest excretory system and secretory system to some extent. Mesoderm in the form of muscle fibers and mesenchyme, parenchyma layer, which we'll talk about that. 3 phyla, which we only study one of them, platyhelminthes, Nemertea, and finally Gnathostomelidia, which we are not going to study these two guys. We only study platyhelminthes. Main characteristics, they're triploblastic animals, bilateral symmetry, I'm repeating myself, flat dorsoventrally, yes, true muscles, they do have true muscles now. It means they have muscle cells, okay? So, incomplete digestive system. What does that mean, incomplete digestive system? We kind of refuse to use it for these very primitive animals. You can say if they have, well, they do not have digestive system, it's just gastrovascular cavity. They do have incomplete digestive system. It means mouth and anus is in the same place. Mouth and anus is in the same place. So if this is your planaria, which you've been doing experiment with it, is that right? So this is the pharynx, the mouth is right here, and also the waste material come out from here. Another nice discussion for dinner table, so you eat more. Oh, no, you don't want to talk about that on dinner table. Some have eye spots, some, not all. Not all platyhelminthes have eye spots. Right here, the 2 eye spots, you saw them on the animal that you're dealing with. They're most monoecious, and the term monoecious, I forgot, can somebody remind me what it is? Another name for monoecious is hermaphroditic, means what? Both sex organs are within the animal. Ovary, and testes, they both are within one animal. The advantage is they don't have to look for a partner, right? I have to look for a partner if I want to have babies. You saw it in the video, but these guys don't. But usually, it's true. Let's talk about it now. They do not have to look for a partner, but in most cases, they find another one, and they call it cross-fertilization. They do exchange sperm. Okay. Why do you want to do that? They have the ability. They have the ability not to look for a partner. But they do. They do cross-fertilization. Why? The question is why? Very good. Genetic variation. Genetic variation for if natural selection, if a disease comes, if a predator comes, at least some of them can escape that disease. Genetic variability. Am I making some sense? We discussed this, Ariana, long time ago. We did discuss it long time ago. I don't know you were here or not that day. Okay, platyhelminthes, parenchyma layer. Most monogenea are ectoparasitic. I will talk about monogenea later on a little bit. They are, monogenea is the name of a class that they are ectoparasite. They're outside of their host, and they cause infestation. Ectoparasites. Ectoparasites cause infestation, and endoparasites cause infection. So if you take your dog to a veterinarian and and the vet says your dog is infected with tick or mite or flea, whatever, you correct them. Say, Mr. Vet or Dr. Vet, my dog is infested with tick or mite or what have you. Infection is inside, infestation is outside. That's a terminology thing that you should know. All trematodes and cestodes are endoparasites. Trematodes and cestodes We are going to study them. So there are 3 classes, 4 classes you have to know: Monogenea, trematodes, cestodes, and Turbularia, which is your platia, which is your Planarian. Indirect and direct life cycle, they can have both, and you all know what direct and indirect life cycle is. Final host is vertebrate animals in most cases. The invertebrate animals that they have to deal with, we will talk about it, snail in most cases. Snails, crayfish, crab, these are all can be as your invertebrate. Triploblastic cnidarians were not. We talked about that. Body fluid moves by muscular contraction. Class Turbularia. Let's talk about that. Turbularia. Edward, I'm tired of saying Ariana and Farhana. They're dozing off a little bit. So, but you're up and ready to go. It seems like you had a lot of caffeine right this morning. So I'm tired of calling them. So I'll call you. Okay, free living, mostly marine. Planarians, of the ones you're dealing with, we do have slide of planaria in the lab on your slide collection. The whole mount and the animal has been cut through 3 regions: through the head, through the pharynx, and through the posterior portion of the animals. You have the cross section, and I'll show you some pictures here. Mouth and ventral sides, no anus. Well, they say no anus in here, but they do have anus. Where the mouth is, also anus is. I hope I'm making some sense. Mouth and anus, same place. Simple life cycles. They have, gastrodermis has phagocytes to engulf food. Class Turbularia continue. Planaria excrete water out. Why? Because they are freshwater organisms. So a lot of, by osmosis, a lot of water goes from outside of the animal to inside of the animal. They have to get rid of the excess water by a system called their flame cells excretory system. Here on this model, they're shown in green. It's a green on this model. On the other model is also green-red type. The other model is a little bit heavy for my thumb, but anyhow. 3 types of neurons: sensory, motor, and association neuron. You study these sensory, motor, and association where in the past we studied it. The reflex arc. You remember that? We talked about this in reflex arc. Yes, in human. Cilia or light-sensitive eye. Asexual regeneration. You cut the animal longitudinally or cross-section, either one. The other half will grow. Some of you did the experiment with that. You cut them in a cross-section. Possibly this end will grow, this end will not. But you never know. You might cut it in a way that both ends grow. I don't know. It depends on how you cut it, but they regenerate. One student one semester said, Amir, it's great you ask questions. I said, what if I put it in blender? If you put this in a blender, what will happen? Will they grow back all of the little? I said, I doubt it. How many cuts? I don't know. You have to cut it and see how many cuts it takes until the whole animal grows back. Again, these are some of the things you can look it up, or we don't know. We really don't know. At least I don't know. How many times, how many cuts, blender. Of course, if you put it in blender, it will not regenerate. But anyhow, Turbularians are monoecious and practice cross-fertilization. I did talk about that. The advantage of cross-fertilization. I did talk about that. Okay, fertilization in female oviduct. That's where sperm fertilizes the egg. They are carnivores. Yes, we've been feeding them raw meat. The metabolic waste diffuses out through the body wall. So if they have waste material, they have a little bit of, on this model or the other model called, here we go, it's called excretory pore. So the green is the excretory system. They have tubes, and I'll show you some pictures, hopefully it comes up. And then they get rid of the waste material through the excretory pore. Okay, so that's one place they get rid of. And the other place they get rid of the waste material is through the anus, right? Mouth and anus, same place, right here. Rhabdoid cells, they are the cells that they are for making the animal wet, fluid, adhesive materials. I hope I'm making some sense. But anyhow, 3 types of muscles: longitudinal muscles, circular muscles, and radial muscles. Of course, on this model, they only show you circular muscles and longitudinal muscles. On the other model, you will also see radial muscles as well. The 3 layers of muscles they have. The radial muscles run this way. Of course, circular muscles run this way. Longitudinal muscles run that way. And then radial muscle, you will see some pictures, and I'll show it to you. Parenchyma cells, they are in the acylomate area. Parenchyma, you learn it where else? Liver, very good. You learn it in liver, okay? And then, and you will see some, I will show you some pictures. And protonephridia, which is the flame cells, make sure in the other model you see. Here we go, great, finally. I'm free now. Pictures in biology says, what? Ariana, how many? Eduardo, how many? A picture says how many? A million, very good, in biology. In other fields, in political science, economics, it's 1,000. A picture says 1,000 words. In biology, says a million words. A million. Okay, brain, they do not, technically, these animals do not have brain. Brain analyze information. These guys, this region does not analyze information. And I'll show you, I got this picture from another source. It's not from your textbook. Your textbook, your lab manual do not call it brain. They call it cerebral ganglion. That's a better term. Collection of a lot of neurons. That's what this is. So anyhow, eye spots, they can detect light intensity. They cannot form image. These animals cannot form image. They can detect the light intensities. If you close your eyes, not put something on it, no. Close your eyes and I turn off the light and on, you will detect it. With closed eyes, you know the light is on or off. So you can detect light intensity as well, okay, with closed eyes. So same as these animals, but they cannot form image. Nerve, yes, they have 2 nerve cords, and then the reason that I want to show you guys that gut, the gastrovascular cavity, the mouth is here. Here is the pharynx. With the pharynx comes out, it's in the middle of the animal you saw. It comes out and act like a vacuum. They suck food. It sucks food, and then of course when it goes inside, it digested by phagocytes right here. The waste material comes back to gastrovascular cavity, and then excrete it out. How'd you like my analogy? How these animals eat? Are you smiling, guys? I can't believe it. She woke up. Wake up, little Susie. Wake up. Okay. Oviduct, of course, testes, they are hermaphroditic, frontal sense organs. Next picture, I think this is in your textbook. These branches of the gastrovascular cavity is called diverticulum. You do have it in your large intestine as well. Those, you have diverticulum in our digestive system as well. So the branches right here is called, the branches are called diverticulum. Pharynx, and at the end of the pharynx you have mouth, of course anus. Here is the food going in, and of course the waste material comes out. Branches of gastrovascular cavity. Here is the nervous system. It's like a ladder. These are called transverse nerves, and these are called nerve cord. And then these are called, right here, they didn't mention it, but in all other animals, this is called lateral nerve on this side. These are transverse nerve, right? A nerve cord, and these on the other side, on outside of the nerve cord is called lateral nerve. Anyhow, the reproductive system, they are monoecious, ovary, oviduct, and testes. We do have it on the model. And then, of course, your flame cells, which are, they grab the ammonia, waste material, more of waste material, and they put it out through the excretory canals and out. Again, I'm showing you the gastrovascular cavity with the food going in through the pharynx. Here are the flame cells. Okay, flame cells, some textbooks say they have cilia, some they have flagella, but they have inside of it, they have flagella or cilia, and then they push the waste material into the duct, right here at the duct, and from the duct, eventually they end up into excretory pore outside of the animal, Okay, that's a planaria. This is a real picture of planaria which you've been dealing with. This is from your textbook. Everything we talked about it. That's a flame cell right there. Everything else here, we talk diverticulum, the branches, ovary, oviduct, vas deferens. Don't worry about vas deferens, seminal receptacles. Don't worry about them. Vagina, all of these little structures. Don't worry about them on the model. But anyhow, I'm looking here. That's what I was looking for. This is the cross section of this through right here, through the pharynx. Okay, we do have slide of this in your slide collection. Make sure you look at them and identify the following. Okay, and then of course you do have another cut through the cerebral ganglion. Oh, I forgot to show you cerebral ganglion slides. We'll go back and I'll show it. Your textbook says cerebral ganglion, right here, it's a lab practical exam question, right here. Not brain, cerebral ganglion. All of that white area is cerebral ganglion. So you have a cut from here, you have a cut from here on your slides, and you have a cut from here, cross section. So let's go over quickly. Rhabdoid cells, as I said, they release mucus on the surface of the animal, and they have cilia here. The cilia aid with the locomotion of the animal. Not all animals have this. Not all animals have cilia, so they can move on them, but generally they have. Then you have gland cells. Again, they release different materials. You cannot see them on your slides. Don't worry about them. Anything like that. Epidermis, absolutely, you will see them on your slides. Okay, epidermis is the skin. Underneath of epidermis, you see a very thin band of circular muscles. And underneath of circular muscles, then these are longitudinal muscles. These dots, you guys see these dots? And then again, on your slides, you see that as well. I have to put them on 40x for you to be able to see that. Okay, when I go here. Then, what else? These